Good morning all. I am Dr. Busineni Goud, Department of Biology, Indus International School, Bangalore. Here I am going to present about DNA profiling of 9 STR loci in Ediga community of urban and rural areas of Tirupati and Chittoor regions of Andhra Pradesh, India. The key objective of this project is to create a DNA database of Ediga population for AP Forensic Science Laboratory. And when it comes to Ediga population hails from Godavari river belt of Andhra Pradesh and Telangana region from ancient times, later expanded throughout the country, and it occupies 17% of population in Andhra Pradesh, comes under backward class. Major occupation was tidy tapping in earlier times, but nowadays they are also doing modern jobs. When it comes to DNA profiling, it was developed by Alec Jeffrey, a British scientist, in 1985, has developed the first DNA profiling technique in the name of restriction fragment length polymorphism, which is based on the repetitive sequences of DNA, which are unique to each individual. Later it was discarded because of its laborious process and most expensive process. STR has been replaced with RFLP nowadays in forensic applications. These are the applications of STR profiling in the case of forensic sciences: paternity testing, historical investigations and missing cases, mass disorders and population genetics. So here we can see identification of Saddam Hussein with the help of their children's DNA also. And these are the different loci present on the chromosomes that indicate the STR markers, and you can see the nomenclature of STR markers also. When it comes to experimentation, samples have been collected from 19 male and 21 female donors from Ediga population with anticoagulant EDTA. DNA extraction was done with the help of phenylchloroform method followed by gel electrophoresis. Amplification was done with the help of PCR. STR profiling was done using ABI 310 genetic analyzer. Results have been tabulated in the form of allelic frequencies. Statistical analysis was done with the help of Promegastat, V12 and software SISA. When it comes to allele frequencies of Ediga population, all different STR markers are showing similarities, exact similarities with the different castes of India spread throughout the country. Here we can see all those castes. When come to other statistical parameters, especially allelic effectiveness, higher values are indicating their contribution towards genetic diversity in the population. With respect to heterozygosity, lower observed heterozygosity values than expected values is showing departure from Hardy-Weinberg equilibrium and a likelihood of inbreeding. Higher values of power of discrimination are making them the best markers for human identification. Paternity index and power of exclusion values are very precise and very high and making them good markers for paternity testing. Also, polymorphism information content is also important in studying the variance of alleles, which has been shown very high values in the case of Ediga population. Probability values, some STR markers have shown greater p values above 0.05 obeying HWE, and some markers have shown less p value deviating from the HWE. And with respect to SD values, very negligible values have been reported, showing little genetic drift in the population. I conclude this presentation by stating that my results not only helpful in forensic application but also useful in population genetic studies, in the study of genetic ancestry of human beings as well as the inheritance of different diseases. Thank you for giving me this opportunity. It's time for question and answers.